Okay, good morning, everyone. Today we have another very important lecture by Dr. Mahadev Rao. He is basically professor of Department of Pharmacy Practice, Manipal College of Pharmaceutical Sciences. He is also the coordinator of Center of Translational Research at Mahi, Manipal. His primary area of research are translational research, pharmacogenomics, precision medicine, and repurposing of drugs in cancer and lung disease. And he has got several publications, and we can see that when he is a coordinator, he must be definitely he must be a good scientist as well. So he has delivered a number of lectures at national and international conferences, and he is also has recipient of several awards. Today the lecture will be on the recorded version, but he will be there at the end to take the question and answer. So what I would like to tell all the participants, please write down your question and answer it in the chat box. We'll take it at the end. So please listen carefully. What is there in the pharmacogenomics in pain medicine or genomics in pain medicine? Genomic research in pain medicine. Probably this lecture will help you to understand this topic. So, Archana, can you start the recording, please?
Good morning, everyone. Glad to be back here again.
Oh, good, very good, Dr. Rao.
Deliver a
Very
lecture on genomic research in pain medicine, which is conducted by CTC Palliative Care Training Program. And many thanks to the organizers giving me an opportunity to talk on this particular subject. Now, why the basic research is important? So this is as per me, natural birth is an accident and natural death is certain. But in between, all happening to the life to death is quality of life. I think to accomplish quality of life, it's important to know the different diseases. Now, since the invention of the human genome, there are a lot of diseases has been characterized very well in terms of the genomic sequences and what are the mutations or the gene expressions are involved in the particular diseases, particularly in the case of the cancer and some other neurological diseases. And now to accomplish better quality of life, it is very important having a basic research to understand the disease, particularly in the case of the pain, where you can translate in the bedside or in the clinical settings significantly. And as you all know that pain management is a human right. I think every physician understand that to treating the patient is how important to understand to give the better treatment for the patients. As Peter, Irish physician who said that medicine is not about conquering diseases and death, but about the elevation of suffering, minimizing harm, and smoothing the painful journey of man to the grave. So now I'm going to talk about how genetic variation leads to the genetic disease. This is a simple example I'm trying to explain here. So there is a gene called opsin, which is present in the X chromosome and location at Q arm, Q28 arm. And this gene is important to have a normal color vision. If this gene is getting mutated, there will be a disease called color blindness, particularly green color blindness or the red color blindness. And you can see this particular disease in 16% of the Northern European descent of Nordic countries such as Denmark, Norway, Sweden, etc. So, so this is a opsin gene here. So it has a different region within the gene promoter. The LCR is nothing but local controlled region. This is in a DNA sequence here, and it has a opsin LW, which is depicted in the red color, and MW1 and MW2 is in the green color. If this gene expresses, you'll see the red cones absent. If the green colored genes are expressed, you will see the green cones present in the IR of these particular people. Now, what happens with these North European descent people, where there is a genetic deletion of some of the part of this gene, for example here MW1 and MW2. So when they get deleted, you have a green cones are absent in the eye, or there is a no gene expression of this, and you get a green color blindness. And in some cases here, for example in the red, you see the gene stop expression in halfway through, and red cones are absent, red color blindness. And sometimes there is a rearrangement or gene conversion, so you have a mix of both red and green here in this case, whereas both MW1 and MW2 genes are okay, but there is a altered properties of the red cones. And in the case of this green colored gene, the first one MW1, and again it doesn't express because of the stop codons, and there is a green cones absent, and they see these particular patients have the green color blindness. And sometimes in the second MW2 gene, it will not express, and it will stop expression in the halfway through. But there is one more copy of the gene is duplicated or expressed, so you could have both red cones and green cones present, and will have a normal color vision. So this is a classical example how a genetic variations leads to the color blindness, particularly the mutation of the opsin gene. So with that, I'm going to put up these contents for today's talk. I'm going to talk about genes and genetic polymorphism involved in the pain. I'll talk a little bit about pharmacogenomics in pain management, and the role of epigenetics in the pain management, and the role of proteomics in the pain management. Then I'll also discuss a little bit about translational pharmacokinetic and pharmacodynamic modeling of pain medications. And I'll also talk about the repurposing of the drugs, which may be very interested in this particular field. And at the end, I'm going to a little bit talk about the basic research areas in the pain and palliative care, what we exactly doing. So now I'm going to talk about the genes involved in the pain perception and modulation. So when skin get hurt by heat, cold, or damage, there is a noxious stimuli will be there. So there are a lot of genes are involved, that is during the transduction. For example, heat-related gene, the TRP1 family members, or in the cold, TRPM8 family members. And during the damage, different genes are expressed. During the mechanical damage, and again the TRP family genes are expressed. So once the signal generated from through this gene, there are a lot of other genes which sent the message to the spinal cord and eventually to the brain, where the pain matrix will be there. For example, the conduction of sodium or potassium channels, there are specific pain genes will be activated. Similarly, in the case of synaptic transmission in the spinal cord, and once it goes to pain matrix in the brain, there are a lot of pain modulating genes are there in the brain, which are acting either centrally or peripherally or in the microalgae, etc. Some of them are well studied, are BDNF, interleukins, NGF, GDNF, and KCNQ. This is the very well studied genes. This is ion channel genes. And the TRP1, which is a cation selective ion channel, which is activated by both heat and red hot chili peppers. That is a capsaicin receptor. So there are a lot of public attention for this. That is how recently got a Nobel Prize for this particular area of research. Yeah, these are the two gentlemen in here, Dr. David Julius and Dr. Ardem, both of them working on the field of neuroscience, where they discovered how we sense the temperature and touch, releases some of the genes, particularly TRP1 gene, and leads to, different messages to the brain. And they also, found the nerve cells sensor for heat, cold, pain, and pressure. And this led to, Nobel Prize for, to these two gentlemen. Now, I'm going to discuss now a little bit about genetic variation involved in the pain. I think there are a lot of different class of proteins are involved in the, pain. For example, enzymes. COMT enzyme, which is in a catechol O-methyltransferase. The decreased enzyme activity is linked to increased pain and increased opioid responses. This is another enzyme, GTP enzyme, where, pain sensitivity correlates with expression of this particular enzyme. And CYP2D6 variants, it's one of the widely studied and used marker in the pharmacogenomics for the pain medications and how they affect the drug metabolism and efficacy. And as I mentioned, some of the ion channels, like a TRP1 class of proteins, the polymorphism change the perception of the pain, and they're the pro-nociceptive. KCNS1, this is another gene, polymorphism is important in the increased sensitivity to neuropathic and mechanical pain. And this is some other, class of the genes which are involved in the polymorphism and involved in the heat sensitivity. And these are other class of proteins, cytokines, for example, IL-10 and IL-6 and TNF-alpha. These are all involved in the one or other way in the pain sensitivity. And receptors like OPRM1, well-known receptor, which is a mu opioid receptor. So if there is hypermethylation of the gene promoter of this OPRM1, there is a lesser gene expression with these OPRM1 receptors, and there will be decreased analgesia. And these are the other two class of receptor ADR, A2 and DET2. And again, they have a different phenotypic changes with the polymorphism. And there are also many drug transporters have been studied in the some of the genes involved in the pain, like DAT1 decreases dopaminergic activity, whereas increases the cold pain sensitivity. And 5-hydroxytryptamine expression, decreases heat pain threshold. And ABCB1, widely studied, drug transporter, which, cross the blood-brain barrier, SNPs, changing the transport activity, thereby changing the analgesic effect. And there are a lot of single nucleotide polymorphism suggested to affect the human pain sensitivity. These are some of the genes here, like GCH1, COMT gene, OPRM1, OPRD1, and so on, TRP1, and CYP2D6, etc. As I mentioned earlier. The interesting thing about these genes are there are multiple SNPs which are involved in the phenotype of the mechanism of these gene expression. For example, GCH1, if you have multiple SNP, there is a partial analgesia. Catechol orthomethyltransferase, there is a SNPs, either increased or decreased pain sensitivity, and so on. These SNPs are very important to determine whether, the patient is eligible to take the drugs which targeting these particular enzymes or the proteins. Now, there are some genes also, used, involved in the heritable pain conditions. For example, HSA and class of protein 1, 2, 3, 4 are there. And if these genes are affected, whether, expression is lost due to maybe genetic mutations, leads to the different phenotype. For example, in this, you can see like a pain and heat loss, developing loss of all the sensation, pain-free phenotype, congenital insensitive to pain, etc. And also there are other genes which are, studied in the similar way. So with that, I'm going to now a little bit talk about the pharmacogenomics in the pain management. It's a very important topic, and pharmacogenomics is a part of precision medicine. In 2015, President Obama, declared, it is a has to be practiced in the clinical settings. And if you look at now, FDA has approved more than 400 drugs based on the pharmacogenomics. And now, if you look at the drug literature, you get a literature in the medicine box. Now, many drugs has to, declare the pharmacogenomics part of the drug. If there is any studies has been done in the clinical settings, they need to declare in that particular leaflets. Now, genomic variation in absorption, distribution, metabolism, and excretion is very important because they will impact the drug exposure. Now, for example, you have an active parent compound here. So if this compound will, go and bind the respective receptors, thereby increase the functional activity of enzyme. Now, what happens is if the inactive metabolites are there, you have a decreased drug exposure. So if you have a normal enzyme, you don't have any, but still you can see, some anticipated exposure, but still you can see an inactive metabolite may go and bind there. And in the case of reduced functionality enzyme, so you have a active parent compounds, and eventually you get a increased exposure of the drug to the target site. Now, in the case of active metabolites, what happens, like a prodrug, for example. So you have a inactive prodrug, for example, morphine to codeine. So if you look at, if they go and bind the target site, and there will be an increased functionality of enzyme, and you have a lesser, inactive prodrug here, you will have a, there will be increased exposure of the drug. And in the case of normal enzyme, as I mentioned, you'll have an anticipated exposure. Whereas in the reduced functionality enzyme, you'll have a decreased exposure of the drug. So this genomic variation again is very important. For example, if you have a normal drug target here, sometimes if the gene, genetic SNPs or the mutation, there will not be any drug binding here. And sometimes you have a variant drug target, there is a possibility of a efficient drug binding. And in the case of sometimes the drugs are, fine, it will go and bind to the target site and efficient drug binding. Sometimes variant drug target, this is a very interesting, you may not have a, binding in here, efficient binding, and there will be a different therapeutic outcome. Now here in this figure, what we are trying to show that the genomic variation is very important in the adverse drug reaction. So ideally what happens is, if a drug which is which has to bind to human leukocyte antigen, where it will be in the antigen presenting cells, it is very important to bind to T cells. If there is an mutation in here or the SNPs in here, it won't bind and there is no T cell activation, and the chances of having a adverse drug reaction is more. Or in the case of you have everything is fine, the drug will go and antigen presenting cell will bind to the T cells and you'll have a T cell activation. And this is very particularly very important in the human leukocyte antigen. And many drugs, undergoes metabolism, through by these mechanisms. And if there is a mutation, you will not have a desired effect. So how is the metabolizing function is determined? So there are three major phenotypes are there in terms of metabolizer. The first one is ultra rapid metabolizer, second one is extensive metabolizer, and third one is a poor or intermediate metabalizers. So in the case of ultra rapid metabolizer, the drugs will metabolize in a faster rate, and in such cases, you require increased dosage of the drugs. And in the case of extensive metabolizer, this particular phenotype where the normal dosage of the drugs is enough, so the patient will respond to the normal dose of the drug. And in the third phenotype is the poor or intermediate metabolizer. And in this case, the patient require lesser dosage of the drugs or the reduced dosage of the drug. So in these three cases, very commonly, seen phenotype in the many drugs, including the pain medications also. But in the case of sometimes what happens is, the patient may not be eligible for the ultra rapid metabolizer because of two reasons. One is he may be toxic to the drug because he has to take increased dosage, and second one may be, there is a possibility of the toxicity of these drugs. And in the case of poor metabolizer, you require a lesser drug, but it may not be metabolized properly, and the drug may be accumulated in the blood, and the pharmacokinetic mechanism will be very poor. So in such cases, alternative medication is important to prescribe to the patients. So this pie chart shows that how pain medications and other drugs used in the neuropathic pain, such as antidepressants. So most of these drugs are metabolized by the class of enzyme called cytochrome P450 enzymes, and they are associated inhibitors, inducers, and substrates that may be prescribed for the pain patients. So there are several CYP enzymes are involved in the metabolism of different drugs by induction or inhibition or substrates. So some of the most important enzymes which are metabolizing the pain medication and others are CYP A3, 4, and 5, approximately 30% of the drugs. And the second one is a CYP2D6, around 25% of the drugs. And the third classification is a CYP2C19 and CYP2C9. Collectively, around 20% of the drugs are metabolized. And more strikingly, a very focused enzyme is CYP2D6, which is significantly metabolizes the pain medication, antidepressants, antipsychotic, etc. So one of the important area in the managing pain in the cancer, very challenging, and it's also time consuming, and lot of trial and error we need to do in this case. Now, opioid analgesics are the drug of choice for moderate to severe pain as per the WHO analgesic ladder. You all know probably, this is the ladder for the drugs which used in the cancer pain. And these are some of the opiates used in the cancer pain: morphine, hydrocodone, methadone, codeine, fentanyl, and tramadol and tapentadol. And these two drugs, I think, not available in India, I guess, hydromorphone and oxycodone. This is particularly marketed in the United States. Otherwise, most of these drugs are available in India to manage the cancer pain. So one of the important enzyme which metabolizes the pain medications are CYP2D6 enzymes. So this particular enzyme is a class of cytochrome P450 family member, and there are approximately 80 known CYP2D6 single nucleotide polymorphism within the coding and regulatory regions of the gene. That includes SNPs, indels, copy number variation, and gene conversion events, etc. So there are, more than 100 characterized CYP2D6 star alleles. Star allele means it is a variant. So each of the star says that it's a new variant. For example, star 1, star 2, star 4, star 9, etc. So such alleles or variants are more than 100 in numbers, and sometimes they have a multiple polymorphisms also. So the genotyping of this particular enzyme is very challenging, but thanks to the technology, now you no need to go and do single SNP sequencing or single CNV sequencing, etc. Now you have a, you can do a next-gen sequencing, or even you can do a whole SNP genotyping, and those technologies are there already to see. So this is very important because we do not know if you have more than 100 SNPs, we don't know which one is involved. So it is very important to know that, which SNPs are important in the different medication. So these are, some of the SNPs which are characterized here, in the CYP2D6 enzyme. It's well studied, but now I think the newer technology will give a better picture. Now, this slide I put up because how these enzymes are there in the different population in terms of a phenotype. So as I mentioned, there are 4 different phenotypes: poor, intermediate, or extensive, or ultra rapid phenotypes. These are the different metabolizers. Now, if you see the SNPs, whatever you see with the CYP2D6 are different in the different population. For example, Caucasians has 6 to 10% SNPs of poor metabolizer phenotype. Mexican American 3 to 6%. African Americans 2 to 5%. Asians are the 1%. And in the case of ultra rapid or high enzyme activity, in this case for the CYP2D6, most interesting to see that Ethiopians have a 30% of this particular genotype and phenotype. Saudis 20%, Portuguese 10%. So what is the interesting? This chart shows that, see, the genotyping of these enzymes are important because what kind of a metabolism they have, what kind of a toxicity and therapeutic effect we have. So if you look at, strikingly, we don't have any data from Indians. Now, if you hear you have a Asians, these Asians basically is a non-Indians. Asians particularly means in this context is the Japanese, Koreans, Chinese, etc. So there is a lot of opportunities to understand in our population whether these drugs works or not. This is one example I'm giving as a CYP2D6 because there is a tons of opportunities to do this research and give the appropriate drug for the patients. So as I mentioned in the previous slide, how CYP2D6 enzyme is important in the metabolism of the, pain medication, particularly opioids. So here I'm giving an example how these enzymes are important. That is what you see in the United States, the opioid crisis, where the #1 selling drug is the oxycodone, which is metabolized by CYP2D6 or CYP3A4. And the metabolism of this drug is important for the therapeutic effect also. So by CYP2D6 you get an oxymorphone, whereas CYP3A4 you get a oxycodone. So these are some of the numbers because the people who take this particular drug in US, it is unbelievable. Around 130 people die every day from this drug overdose. This drug overdose may be due to either poor metabolizer or maybe rapid metabolizer. We don't know. There is no such testings are done frequently. Around 10.3 million people misuse these drugs. And the drug which was invented by the Purdue Pharma, I think the CEO and all of those people in the jail now, I guess, or maybe they have come out because there is a big conspiracy of selling these drugs in the country to people who really don't want, because this particular drug is useful in the palliative care setting rather than the normal one. And I was a pharmacist there. I remember kids they get the fake prescriptions and take these drugs, and many people die also. So these are the numbers you can see here. It is very sad that how this misuse of the drugs. But In the scientific context, I think pharmacogenomics is very important. As I mentioned, this drug is metabolized by CYP2D6 and CYP3A4. So it is very important to know that the status of this single nucleotide polymorphism genotyping, whether they are eligible for this drug or not. So what are the clinical consequences for this poor metabolizer and ultra metabolic phenotypes of CYP2D6? Because I'm focused on only this enzyme because, as I mentioned, this particular enzyme metabolizes majority of the opioids. Now, if you look at here, in the case of codeine, if it is a poor metabolizer, failure to respond, poor analgesic effect. So you give the just drug, there is no outcome in terms of what you are expecting. Similarly, with the tramadol and also opiates, and these are the three drugs which are in a poor metabolizer. So you may not find the better therapeutic outcome. Now, in the flip side, what happens is when you take these drugs, if they don't have a therapeutic effect, you keep taking this drug. You know what is the side effect? One of the biggest problem with these drugs are the respiratory depression. That is why, as I mentioned in my previous slide, in the opioid crisis, many people die because of the respiratory failure. And in the case of ultra rapid metabolizer, there is an increased risk for toxicity because you are taking more amount of the drug or increased dosage of the drug. If you keep taking the codeine, and you'll have a morphine toxicity. Again, side effect is the respiratory depression or respiratory failure. Now, interesting thing is that, these opioids, basically metabolized by the phase 1 or phase 2. And very important point to note that the drug-drug interactions that might lead to unpredictable clinical response. So if you look at, these are the different opioids here. They give the codeine and hydrocodone. It will be phase 1 metabolism by the CYP2D6 enzyme. No phase 2 metabolism. You have a potential to have a drug reaction is based on the grade, and it is a potential to have here drug interactions, and very highly likely to have a toxicity or poor therapeutic outcome. And in the case of tramadol or oxycodone, both CYP2D6 and 3A4, as I mentioned before in my slides, oxycodone or tramadol goes to metabolism by the two of the enzymes. Now, no phase 2 metabolism, potential drug interaction is very high. Again, toxicity or poor analgesia is high. Fentanyl, CYP3A4, same thing. You'll have a potential to have a drug interaction is very high, but lesser toxicity or maybe lesser therapeutic outcome. Now, interestingly, morphine is probably more safer in that sense because it doesn't go under phase 1 metabolism. So it does go with the phase 2 metabolism, glucouronidation, and via the enzyme UGT2B7 enzyme. So you have a lesser drug interaction or lesser toxicity or poor analgesic effect. So what I'm trying to say that, when you give these drugs to the patients, most of them are palliative care patients, and these people will have already many drugs with the comorbidities, may go through again some of the metabolism using this enzyme. So it is very important to understand drug-drug interactions in these particular patients. And again, if you look at geriatric patients, I think most of the palliative patients are maybe geriatric patients, I guess. And these patients again have a more drugs which they are taking for the comorbidities, and they may not get any benefit by giving these drugs. So it is very important to understand how these drug interaction takes place. That is why I always suggest that the role of clinical pharmacist in here is useful. Usually In the United States, they have a clinical pharmacist. They are trained in the palliative care settings. They know how to give the drugs to geriatric patients and palliative care patients. And some of the pain management drugs is involved in the adverse drug reactions. Now, pharmacogenomics studies might help to reduce this. For example, anti-epileptic drugs for neuralgia like carbamazepine, phenytoin, lamotrigine, clozapine, and all of these induces the cutaneous adverse drug reactions such as SJS or TEN syndrome. This is basically due to mutation in the human leukocyte antigen A or B. So when there is a mutation, the antigen presenting cells would not bind to the T cell appropriately, and thereby having a adverse reactions are very common. If, for example, here some of the polymorphism genetic variants, particularly HLA-B1502, very predominant related to this critical event, and by knowing these genotyping of these HLAs, it is easy for the clinicians as well as clinical pharmacists to give the appropriate drug. I'll give an example here. This is basically an anti-gout drug, allopurinol, which is given to a patient, and he ended up having you can see here in the picture, we published this particular paper in the hospital pharmacy, and where we have seen, hyperimmune reactions. And this particular patient was lucky to come to our hospital, and we realized that the way we saw the hyperimmune reaction or Stevens-Johnson syndrome or TEN syndrome, so we asked him to give the sample of blood, and we isolated the genomic DNA from the peripheral blood lymphocytes, and then we sequenced for the HLA-5801, and we found this particular patient had this particular SNP in the HLA-5801. And then we suggested this patient not to take these drugs in the future, and also we suggested for his entire family not to take the medication before you test for this particular genotyping. So now there are, a lot of guidelines for pharmacogenomic based prescribing. So there is an international consortium called CPIC, or Clinical Pharmacogenetic Implementation Consortium, and also PGRN, which is a part of NIH, and PharmGKB, etc. And these are all endorsed by the American Society of Hospital Pharmacists, Clinical Pharmacists, etc. Now they have a consideration for assignment of the CPIC levels for the genes or the drugs, and there are 4 levels are there: A or B, C or D. In the case of A or B, prescribing action is recommended. That means alternative therapies should be suggested in this case. If any drug comes under this classification, you need to change the dosing and the drugs as well. In the case of if level C, no prescription change required based on the genetics, but alternatives depend upon the physicians and the clinical pharmacist. Whereas for the level D, you don't require any change of the prescription, so alternatives may not be required, and because it's depend upon the what evidence you see. So these are some of the pain medications which used in the practice. For example, amitriptyline, it is a tricyclic antidepressant. So if the level is A, CPIC level is A, they need to screen CYP2C19 and CYP2D6, single nucleotide polymorphism. So in the US, it is a mandatory. You need to, screen these genetic variation for that patient, whether that SNPs are there or not. Similarly, with the celecoxib, codeine, methadone, and so on, it's a depend. Again, for example, celecoxib, you have a CPIC level B, so you need to, you can do or cannot do. It's up to the patient whether he wants to do CYP2C9 or not. But in the case of level A, it is a mandatory to do a SNP genotyping. Switching the gear to pharmacogenomics to epigenetics. So there is a lot of interest now to drugging the pain epigenome. So epigenetic mechanism is nothing but hypermethylation of DNA, histone modifications, particularly H3 or H4, and microRNAs, which strongly affect the regulation of the gene expression. And there is a lot of research showing that they are associated with the pathophysiological pain. And several studies, mostly in animals, shows that inhibitors of DNA methylation and activators and inhibitors of histone modifications and miRNA modulators reverse the number of pathological pain epigenome which are associated with the altered expression of the pain relevant genes. And the epigenetic modulation is very important. So they may reduce the nociceptive response and provide a novel therapeutic option for the analgesic therapy, particularly in the chronic brain stress. So what is the epigenetic mechanism? So you have a histone tail which is attached to the chromosomes. So there is a lot of epigenetic mechanism like acetylation, histone deacetylation, methylation, and histone demethylation. These are some of the events happen in the chromatin. And when these events happen, either gene transcriptionally active or inactive. For example, acetylation will lead to the transcriptional activation of the gene, whereas lysine 4 methylation of the gene will be again transcriptional activation of the gene. If histone deacetylated bind here or histone lysine trimethylation of 27 of the histone H3 leads to the transcriptional inactive genes. So this is a very important mechanism. Sometimes these histone modification is also associated with the DNA methylation. So DNA methyl transferases which methylates the cytosine of the DNA. So these CPG dinucleotide clusters are called CPG islands, and when these CPG islands are present in the DNA, there it is called as a hypermethylation of the DNA. So combined with the histone modification, there is a significant research showed that epigenetic silence of the pain-related genes. So these are some of the pain-relieving compounds that exert epigenetic effects based on the literature. For example, celecoxib is an anti-inflammatory drug. So they showed that in the inflammation and musculoskeletal inflammation, there is a epigenetic linkage has been established. Similarly, with the opioids for the cancer pain, acute pain, and hyper analgesia, etc. Fluoxetine, amitriptyline, valproic acid in a neuropathic pain. Valproic acid is one of the most interesting drug. They have shown that this particular drug as a histone deacetylate inhibitor, and there is a lot of, I think, tons of studies have been done in the epigenetic area where valproic acid is a very good histone deacetylate inhibitor. So this is a table which shows that some of the drugs, for example, HDAC inhibitors like trichostatin A, urinostat, they showed in the preclinical level how they involved in the nociception mechanism, and they also show that there is a pain relieved by these drugs in the preclinical studies. And there is a couple of drugs like givinostat and romidepsin. They are in the phase 2 trials. They also involved in the pain relief by some of the genes which are involved in the pain. And another two interesting DNA methylase transfer inhibitors, pi-azacitidine and decitabine, and they showed that the clinical study shows that they are relieving the neuropathic pain in the rats or in the mice mediated by some of the genes related to pain. For example, is the OPRM1. So other than DNA methylation and histone modification, there are a lot of miRNAs are involved in the mechanism of the nociception. So these are some of these miRNAs expressed in the spinal cord. For example, MIR146A, the target gene is a TRAP F6, which decreases this particular gene. MIR195 for ATG14, and MIR183 is a BDNF, which this particular gene is a peripheral modulator for the central nervous system. So another area in the pain management is a proteomics, and also the newer one is a metabolomics. So there is a lot of studies, are people doing to understand how the proteins are important in the pain management. This is very important because, in the genome, always you see the mRNA expression, but sometimes it may not be translated in the protein level. That is why this proteomics is very important to understand the basic research where you can see whether, protein itself is expressed, or sometimes, post-translational modification is also important. So, proteomics techniques are, it's a very advanced, and it's a very expensive techniques to do, real-time research, and the instruments what you require, like a mass spectrometry, liquid chromatography, mass spectrometry, etc. But these all will give, translational and post-translational modification, whatever which is associated with the persistent pain. And identification of these proteins are important, so you get or you'll reveal a novel therapeutic targets. And there is a lot of preclinical and clinical research going on in this, understanding the role of proteomics and metabolomics in the pain. So another area is a translational pharmacokinetic and pharmacodynamic modeling of pain medication. So as you all know that, PK is the study of the mechanism of drug absorption, distribution, and elimination, and the kinetic of this process. And pharmacodynamic is nothing but study of the biological effects of drugs and their mechanism of action. And now, PK/PD modeling is used because in the traditional pharmacokinetic model, you have to remove or you have to collect the blood sample from the patient maybe 10 or 15 times. But the newer population pharmacokinetic or mathematical modeling, you do not require taking the samples for multiple times. I think 2 or 3 times if you take the drug, you can do a mathematical modeling, and this will be definitely helpful in the case of the pain medication. Taking a blood sample for 10-15 times, it is very difficult. So that this is the new area where population pharmacokinetic modeling and pharmacodynamic modeling will be useful in the pain research. So this schematic overview shows that how PK/PD modeling is done. So you have a patient population, give the drug and measure the plasma concentration, see the efficacy and toxicity, and also you need to see whether treatment adherence is there or not. Then you will study both pharmacokinetic and pharmacodynamics. Then find out what are the contributing factors like a demographic, lab values, laboratory values, pharmacogenetic, like as I mentioned, SNPs, whether they are involved in the some of the genes which are metabolizing the drug, for example, CYP2D6 in the case of morphine metabolism, etc. And then see volume of distribution in the different tissues in the body, like a lung, heart, liver, etc. And then find out the pharmacology part, what is the concentration of the drug in the blood. And finally, derive a mathematical modeling where you can give the appropriate dose of the drug. And this particular now branch is called as a pharmacometrics, and this is a part of precision medicine now. And these are some of the drugs, they already have done a PK/PD modeling. For example, pregabalin, morphine. Pregabalin basically for the treatment of fibromyalgia, and they also did for the morphine for the pain relief. How dosing is given in the post-operative settings, and again in the case of surgery, how morphine is used by this pharmacokinetic and pharmacodynamic modeling. And fentanyl, this basically very interesting. They characterize the PK/PD correlation of intranasal and intravenous fentanyl in opioid naive patient undergoing third molar extraction. It's basically used in the oral surgery. So there is a lot of research going on in this area also. So I'm in my last part of the talk, I'm going to talk about repurposing of the drugs. It's a part of the precision medicine now. So repurposing of the drugs, nothing but these drugs are already approved by the FDA. And in the traditional approval of the drugs takes place 15 plus years. In this case, we know already the preclinical studies, we know the toxicity, we know the pharmacokinetics, we know majority of the research has been done. So that is why this might take very lesser time. Probably you may have to go to directly to the clinical trial, or maybe these are some of the rules for repurposing drugs. So you need to know what is your target. Then you go and do an in silico screening, either ligand based or the docking. So there is a software called Schrodinger software, where this software will tell whatever the gene of your interest, whether this particular drug will target or not. And then you collect the patient data like omics data, and then you collect the drug omics data, then you prepare the library, all these data you put it together, and then you can take this particular drug, either you reformulate it, or you can we traditionally do, for example, sustained release or prolonged release, whatever you want to do. Then eventually you can do the clinical trial, the disease which you want to try. So in this case, there are more than 3,000 drugs are there, and there are more than 7,000 drugs which are not used in the practice. All these things, we have a library. We can go and screen the gene which you are interested in the pain management. There are a lot of genes, as I mentioned before. We can go and screen, and we can see whether these drugs will be inhibited or targeted those drugs. And with that, you can go back and do the research in the preclinical as well as in the clinical settings. And I think it's a very interesting area. I think a lot of drugs are out there. That's what we call off-label uses of these drugs. I think you all know there are many drugs are there, for example, minoxidil or sildenafil citrate, etc. So these are some of the example, for the repurposing of drug. So I'm going to focus on one of the drug here, duloxetine. So this basically used as an antidepressant, I think selective serotonin receptor inhibitor, but it is basically used as an antidepressant and anxiety, but it is also used in the neuropathic pain and to treat urinary incontinence. That is what, repurposing of this particular drug. So these are some of the potential basic research areas in the pain and palliative care in terms of, pharmacogenomics and population pharmacokinetic. So we have a project on the tapentadol we are doing here in Manipal in our patient samples, and developing a PKPD models of opioid drugs such as morphine, tramadol, etc. So we have written a big grant with Dr. Naveen Salins to test in Indian population. And third one, pharmacogenomic integrated drug metabolism, drug-drug interaction studies of pain management drugs. So we are doing now some, retrospective study, what all the drug interactions are there. And again, this is the most important one, whole genomic, epigenomic sequencing and proteomic or metabolomic sequences to identify novel biomarkers for pain management, as well as understanding the mechanism of pain management drugs in Indian population. Remember, most of these drugs, what we have now is, done clinical trial or clinical research in the Western population. So till 2018, we didn't have that mechanism to try in our population. I think there is a lot of opportunities, in this area where we can do basic and translation research. And finally, repurposing of the drugs and developing novel molecules for the pain management. I think there is a tons of drugs in the library. We have to screen, and we have to see which target you want to see in the research for the pain management. With that, I'm going to end my talk, and thank you very much for listening. If you have any question, I'm very happy to answer. And this is my phone number here. This is my email. You're all free to ask any question in case. Thank you again. Thank you very much.
Thank you very much, Professor Rao. It was such an important and you have explained it in a very simple way. I think those who are listening, they must be really getting motivated that we should definitely, there is hardly any data from Indian setting and they would like to uh, start working on this area as well. So there is a question from Dr. Shrikant. Dr. Shrikant, can you open your mic and ask, please? Shrikant, Dr. Rao, you are here?
Yes, madam. Thank you. Thank you very
Okay,
much.
thank you very much.
Thank
It was such an excellent presentation.
Yeah,
Shrikant?
thank you.
So basically, he's asking about the finance part, cost implication of two patient once implemented in Indian setting. I don't know what he wants to ask in this.
Yeah. I think it's a good question because many a time people ask, this is a very expensive. They don't have money to get the medicines or the regular treatment. But if you look at, the money what they spend otherwise is more expensive than sequencing. Now, I mentioned at one point of my talk, for example, CYP2D6, which is, majority of the pain medications are metabolized by this particular gene. And if you look at more than 100, SNPs are there in this particular gene. But if you now, technology is very advanced. If you see the next gen sequencing, you can do all of them or now whole SNP genome sequencing. It costs around 25,000 rupees. But if they do, it is more beneficial than the drug which they are taking and eventually no benefit at all. For example, opioids, if you give, the side effects are particular respiratory depression is a big deal with this particular class of the drugs. I think instead of you are taking the drugs which don't work, and also in the palliative cancer care patients, drugs are very expensive. So we always say that, these sequencing may helpful to the patient in a way that they can get benefited better than the, if you are not doing the sequence. It's a good question. I know Indian setting, many people ask, oh, it's very expensive, 5,000, 10,000. It's a big deal for the poor people, I understand. But still, it is beneficial if they do it. That is my personal. I told you one example of allopurinol, the drug which the patient taken. So he survived actually, and his whole family now sequenced. So it will get, if they have a polymorphic SNPs, there is a possibility that their family members also may have. By sequencing, there is a lot of benefit, not only to them and other people too within the family. Thank you.
Thank you, Rao. Thank you, Professor Rao. Thanks a lot. So, are there any question? Anybody wants to ask anything? Anyone? Anyone has any comment, please? So, comments and questions. Acha, there is one. Let me see what is the question. Okay, there is no.
Good morning, sir. I wanted to know how did we get the percentage of polymorphisms in various continents? Have we tested enough patients to? Have we tested enough people to know the percentages exactly, or among the tested patients we have checked the percentages?
Well, I think end of my talk I had written that statement. We have not done genomic sequencing in our population, either whole genomic sequencing or epigenomic. There are a lot of projects which is conducting now by NIH, maybe 1,000 genome projects. They do for most of their diverse population. So that is the one important thing. Actually, I think even last time when I presented, I was talking to Dr. Bhatnagar, the same thing. We need to push our population to get the sequencing done, so person-based or in the population-based. Then we understand better what kind of sequencing will be there in our population. Now, again, this sequencing or SNPs are, maybe different in the different ethnic groups or populations. So we don't have much study in India. So it is a time where, we could do, especially younger generations who can, look into the future in this area. I think you can sequence those, some of the genes which are already established, or we may see the novel novel SNPs in our population also. So now personalized based, very few people do, but I don't think so anybody does in India. But in the United States, I think in many hospitals, there is especially Mayo Clinic, you can see they have around 800 plus drugs they are doing, SNPs for the different, drugs for the different population. So in India, we are not yet there, but I think it's a big opportunity for us to start.
I think it is 7:30. We will stop. We'll have to stop. But
Yeah.
thank you for giving your time, and it was such an important and very, very clear and crisp presentation. Thanks a lot. Thank you, everyone.
Thank you, Dr. Bhatnagar. I think I'm glad to see again you all, and I think in future also, if you are in this area, you want to collaborate or explain the things, I'm very happy to do. Thank you very much.
Thanks
have a great
a lot.
Monday.
Have a great Monday, everyone, and have a next week, and next week Monday morning 6:30 we will Dr. Jennifer will speak how to write manuscript. So please don't miss to join the next Monday morning before 6:30. Thank you very much. Thanks a lot. Thank you very much, Professor Rao, once again.